So I come into the National Academy of Sciences as an engineer in the engineering sciences section. And so my background is mechanical engineering. About 10 years ago, while I had already migrated from mechanical engineering into material science, about 10 years ago, one of my colleagues came to me. I was head of the material science department at MIT, came to me and he said, "Why are you studying dead materials? Why don't you study living materials?" So we thought, what a great idea. Why don't we take approaches and ideas and models and theories that we apply to synthesized materials and see how far we can take them with respect to living systems using new tools and technologies. So you heard about in the first talk collecting fecal samples, 9,000 fecal samples from monkeys in Africa. And so rather than going to the jungles of Africa to do that, we also studied malaria by going to the jungles of Cambridge, Massachusetts, and collecting different types of samples from an engineer's perspective. So that's what I'm going to talk about in the next few minutes. So coming to study human diseases and looking at the scientific mechanisms or mechanistic processes that underlie human diseases, but with an engineering perspective. So that's what the title reflects. So the motivation for what we have tried to do is the following. Coming as a mechanical engineer with a physics bent, the basic question we ask is the following. Take any broad disease class that you want to focus on, whether it's an infectious disease like malaria, a hereditary disease like sickle cell anemia, or a disease whose origins are relatively unknown like cancer. You take any one of these and ask the following question. As the disease progresses, at the cell and molecular level, you go from a completely healthy state to a completely pathological state. What are the systematic changes in the physical properties at the cell and molecular level as a result of the disease? And that question, surprisingly, to my knowledge, has been answered for most of the diseases, major disease classes. So that's what we set out to do. You can also ask the inverse question. If something comes to the human body from an external medium or an environment or a vector like malaria, or if something happens, or pollution, or something happens naturally in the human body, how does it systematically lead to changes in physical properties, and how do you go back to a disease state? So the first question, if you're not in the field, the first question you ask is, why does it matter? So here is a simple illustration of this. Every second, our bone marrow produces hundreds of thousands of red blood cells. So a red blood cell is a disc-shaped, and it has a long diameter of 8 micrometers. So just to put it in context, 8 micrometers is about 1/10 of the thickness of the human hair. Okay, that's the size of the red blood cell. We have billions and billions, tens and hundreds of billions of red blood cells in our human body, and that's why the color of blood is red. The red blood cell has no nucleus, so it circulates through the body approximately for about 120 days to deliver oxygen to distant corners of the body and to take carbon dioxide back to the lungs. So its function is mainly a delivery service, FedEx for delivering oxygen and taking back carbon dioxide. Where does mechanical engineering come in? So the inner opening of a blood vessel in our brain, a tube, a flexible tube, is about 2 to 3 micrometers, which is roughly 1/3 of the large diameter of a red blood cell. So an 8 micrometer red blood cell has to squeeze through a 3 micrometer diameter tube to deliver oxygen to the brain and to take carbon dioxide back. If it doesn't do its job, the brain won't get oxygen. Disease happens. Conversely, if the ability of the cell to stretch is compromised, some disease is going to happen no matter what the reason is that led to that compromise. So that's the motivation for this study. So we studied over the course of the last decade or so 3 different diseases. Plasmodium falciparum malaria, which you heard about, but from a nanotechnology perspective. Hereditary blood disorders like spherocytosis, elliptocytosis, sickle cell anemia, and different types of cancers, leukemia, pancreatic cancer, and breast cancer, and so forth. One of the things I have to say with respect to my background is about 10 years ago, I decided to immerse myself into a very different community. So rather than going to the jungles of Africa, I went to Paris. And inside Institut Pasteur in Paris, in a group of MDs, hematologists, biologists, microbiologists, and that's where I picked up most of this, what I know today. So let me first start with the first question and a first demonstration. Physics and engineering meet cell biology. So one of the longest known problems in cell biology is theoretically known, is all living cells flicker or fluctuate or vibrate. But until a few years ago, until this work, nobody was able to measure that for the whole cell. Here is the reason for it. The magnitude of vibration is of the order of a nanometer. The diameter of the cell is 1,000 times bigger, is of the order of micrometer. And the time scale of vibration is a millisecond. Plus the cell is in a fluid, and if you use physics or lasers, it complicates life. So how do you measure it? So we decided to do an experiment to set up a technique. So since we are at the National Academies and the editor-in-chief of PNAS is here, so I have to show this. So this is a result from one of my graduate students, Paul Park. And the experiment we did was very simple, but directly borrowed from physics to address two issues. If you take a cell that naturally flickers, without contacting the cell physically, can you measure the magnitude of flickering for the whole cell in real time, non-contact, so that if it doesn't flicker by the right amount, can you diagnose a disease? Conversely, if you know the disease, can you estimate how much less or more it'll flicker? That's the first question. Second is, what is the connection between chemistry and flickering? For example, ATP is an energy currency in the human body. If you add ATP, or if you take ATP away from cells, will the flickering increase or decrease? And how do you measure it? And that had not been done until this work for the whole cell. So here is how it works. I know people here, there are people here who are not scientists or engineers. There is a rule in giving lectures to a general audience, you don't show equations, especially after lunch. Unless you are an engineer, you're allowed one equation. So I have only one equation here. So it's a very simple technique. So what you have in the middle of the figure, that there is a red blood cell. The red blood cell refracts light. So it has a refractive index inside, and there is a refractive index outside the cell. When you send a plane wave of light, like a laser beam, because of the difference in refractive index between inside the cell, which is hemoglobin, and outside the cell, the light undergoes what is called a phase shift. So when it comes out on the other side, you see the phase shift. It looks different. And that phase shift can be automatically captured in real time by a computer by that complicated equation, which is an E, and on top of it is something else, delta phi. The delta phi characterizes the phase shift. So if you know the wavelength of light, which is lambda, that's the one simple equation. H is the thickness of the cell. Lambda is the wavelength of light. Pi is, of course, 3.14. Delta n is the difference in refractive index between inside the cell and outside the cell, which we know. And delta phi is something that you measure. So you know everything you need to know. So you measure the thickness of the cell to a resolution of a nanometer to a time step of a millisecond for the whole cell, problem solved. Non-contact, just with a simple laser light for a short period of time. So here is how this works. Let me show a video, first for a healthy cell. Sorry. So first you're going to see a healthy red blood cell, see how it flickers. This is from the experiments. That's the time step in millisecond. This is a nanometer scale experiment. Now you put a Plasmodium falciparum malaria parasite inside there, ring stage. Just after a few hours, it still flickers quite well. But if you wait about 24 to 36 hours, in the late stage, the flickering is much less uniform. So using this technology, non-contact, you can tell what pathological state you have for the cell in real time at a using nanotechnology and millisecond time step. Another example, so there was a Nobel Prize given in 1997 to 3 people. One of them was Energy Secretary Steve Chu for studying optical properties and laser properties, optical trapping. And this is an experiment taken from physics, but in the context of malaria. So in your top left, you have a human cell that's connected to 2 glass beads that are attached. You shine a laser beam on the glass beads, which are like handles, and you try to stretch the cell to measure how much it deforms. The job of the red blood cell is to deform as it moves through the body. So in this top middle figure, you have the same cell, but we impose a force of 68 piconewtons. So for those who are not scientists in the audience, if you take an average size apple in your hand and stand on Earth, the force on the hand due to gravity is 1 Newton. This is 10 to the minus 12 times 1 Newton, tiny, tiny force. And we can put that in a controlled amount. If you go to 150 piconewtons, you can see how the cell stretches. But if you now take the same healthy red blood cell, put a malaria parasite inside there, as you see in the bottom row, at all forces, the cell refuses to stretch because the parasite transports protein to the cell, and the protein interacts with the cytoskeleton of the cell becomes very stiff, it loses its ability to stretch. That is a mechanical phenomenon. That combined with the fact that cell also gets very sticky in the body during malaria, just in 48 hours, the increased stiffness and the increased stickiness makes it more difficult for the cell to move through the body and therefore to deliver oxygen to different parts of the body. In the very advanced stages of malaria, you have cerebral malaria, which is what causes fatality due to brain not getting oxygen, or placental malaria in pregnant women. Now, this is all mechanical engineer's view. Now let's go to genetics. What does it say? So based on experiments that Institute Pasteur did in Africa, it was suspected that there was one protein that probably causes this increase in stiffness. And this protein was suspected to be something called RISA, which stands for ring-infected erythrocyte surface antigen. So the question is, can we use all these tools to understand if RISA causes the increase in stiffness that I just demonstrated for you? So here is the experiment we did. We were fortunate. I was in Paris in 2004. The Human Genome Project was done in 2001, 2002. In 2004, the genomic sequencing of the Plasmodium falciparum parasite was figured out. So now what we could do is we could take a parasite, genetically clone it, and knock out just the RISA protein. We can not only knock it out, we can knock it back in. That gives us a beautiful control experiment. So we did the same experiment that I showed you in the previous slide. In the top figure, you have 3 colors. It's a triple immunofluorescent micrograph. Red is color-coded for a protein in the red blood cell that we all have. Blue is the parasite that you see inside the cell. So every red blood cell that has a blue parasite inside it turns green because the parasite exports the RISA protein to the cell, and the RISA is color-coded green in this figure. Now we take the parasite, clone it, knock out the RISA protein, put it in the cell, do the same experiment. The bottom right figure has no green because we intentionally knocked out the RISA protein. All it proves is the knockout is successful. Now we take both of those and do the same experiment, and this is what you get. So on the left, 37 degrees Celsius is the normal body temperature. The left side there, where it says 10, that's the wild type, or that's the healthy cell. The middle one, the gray one, is the wild type that has the RISA protein in it. So when you add RISA protein, the stiffness of the cell increases 3 to 4 times, just in the first few hours of the parasite being inside the cell. Now, if you knock out the RISA protein, just the one protein, the stiffness drops. That's at body temperature. Now, if you go to febrile temperature, you know that when somebody gets malaria, every 48 hours they have a fever episode. So what happens is that when the body gets the parasite, the body tries to fight it by raising the temperature, but the RISA protein and the parasite fight back because RISA stabilizes the cytoskeleton of the red blood cell. By knocking out RISA, you prevent the parasite from having the upper hand in that battle. So this result conclusively shows that RISA protein is responsible for significantly increase the stiffness, but if you knock in RISA, the stiffness goes back up. So it's a conclusive experimental proof of this. This someday can lead to a potential avenue to knock out RISA, either in the mosquito before it gets to the human body, or in the human body through some therapeutic treatments. Now I'm going to close with a few, see if it moves. Can you advance by one, please? Next one. Okay. So this is all static. How do we know what happens in the brain as blood flows through the brain? MRI just will not do it. It doesn't have the resolution. We can't do it on humans. So we can do in vitro. So we borrow technology from semiconductor manufacturing, a microfluidics technology. And what I'm going to demonstrate to you in this video clip is a channel which sort of mimics a blood vessel in the brain. So the inner cross-section is very similar to the inner cross-section area of a blood vessel in the brain. Of course, this doesn't have any chemistry in it. So we're going to send healthy red blood cells through this, and they're going to come out here, and you will see what happens. So this sort of visualizes blood flow in the brain dynamically. So this is how red blood cells go through blood vessels in our brain. So this is made of a polymer, a relatively flexible polymer. So this is a healthy red blood cell. Now suppose you have a red blood cell that has a malaria parasite in it. My hypothesis is that this parasite will make the cell very stiff, and because of the stiffness and the stickiness, the cell either will block an entrance to a microvasculature, or it won't be able to squeeze through. Let's see if that is true. So the next slide demonstrate this. So this is a collaboration with a group in Singapore at the National University of Singapore. So what you see is a long, like a cocktail glass. You have a long stem, and the diameter of the stem is the smallest blood vessel. It mimics the smallest blood vessel in the brain. At the entrance to the stem, you have two cells. This is a red blood cell with a parasite here. This is another red blood cell with a parasite here. All the others are healthy red blood cells. I postulate that these two cells are too stiff to go through here, but the others should be able to go through to demonstrate the effect of malaria parasite. So let's see if that works. So these two are stuck, and you'll see in a minute as we add more red blood cells. So instead of 2 now, if you had 20 of these blocking an entrance, you will have a problem delivering oxygen to the surrounding tissue. So next question is, can we use all this knowledge to develop diagnostic tools? For example, ideally, malaria happens in developing countries, in Africa, in places like Thailand, Indonesia, and so forth. In remote areas, where rather than a technician taking a blood smear, sitting in front of a microscope and clicking the number of infected cells, can we automate it? So if I could do anything, can I have a chip about the size of a thumbnail, and I do or a technician does a fingertip needle prick, takes a drop of whole blood, puts it in the chip, and does the simple microfluidic experiment. It's a portable, disposable, inexpensive tool which costs, let's say, 50 US cents a pop, and you can throw it out after the experiment. Can we answer the following questions? Does the person have malaria? Do they have Plasmodium falciparum malaria? Do they have Plasmodium vivax malaria? Because vivax is not as lethal as falciparum malaria. Do they have fever because of malaria? Can we filter out the number of infected cells from the uninfected cells so that we can automatically compute parasitemia? So these are all questions that are within the realm of current technological possibility. Can we demonstrate it? So here is one demonstration. So we create an obstacle course for red blood cells. So different shapes, diamond shape, converging, diverging. We send a whole bunch of red blood cells, and we've demonstrated that with whole blood as well. And we ask the question, depending on whether they are infected or not, and depending on the stage of infection, the cells move at different speed. From the speed with which they move, and from the high-speed photography, we can tell whether it's a healthy cell or an infected cell. We can compute the number of cells automatically and very quickly, and we can answer a lot of questions. So the technology exists, and in this work, we demonstrated this. And you can see how the cells go through, and infected versus uninfected will have a very different response. And during the 48-hour cycle, we can systematically map the progress. So this is something that's within today's realm, but it'll take quite a bit of development to bring it down to about 30 to 50 US cents a pop. So going from experiments to computations, this gives us a unique power to use computational modeling, not just for one or two cells, but hundreds of thousands of cells flowing through blood vessels. How can we visualize them through computer modeling? So in the top is an experiment done at University of Washington in Seattle, where you have a channel, and you put only red blood cells, and some of the cells are infected. You can see the little black dots inside them. They're infected with Plasmodium falciparum. So if you look at this figure, that's how blood flows. You can see the rolling of the cells on the blood walls, the channel walls. Now, if you use atomic level simulation, it's too fine. It's not useful to do thousands of predictions. If you use continuum models, it's too coarse. You want something in between. So we developed a technique for biorheology in collaboration with Professor Carnegie and his group at Brown University to do large numbers of cells using a technique called dissipative particle dynamics. And here is a demonstration of what the computer simulation does. Now, we can not only do red blood cells, we can do white cells, plasma, platelets, etc. We can do whole blood and answer a lot of questions that could not otherwise be answered until now. We can do adhesion. Now, how do you go from cells to organs? So the discovery of the HIV thing came from a spleen in a freezer. Human spleen is a filter. So there is a group in Paris, our collaborators, Pierre Buffet and co-workers. They get human spleen from pancreatic cancer patients who need splenectomy. The spleen is still good, and it's outside the human body, so you can run experiments ex vivo. So they do experiments with the infected and uninfected cells. They also developed a synthetic bead system to simulate the human spleen. We do our computer models here, and using those models, you can decipher how cells go through the human spleen. So right now, the engineering of that is not understood, and this gives us a unique vehicle to do that. And this is my last slide. This is all infectious disease. You can also use these tools to other type of diseases. I'm going to show just one result for a sickle cell disease. We've done similar things for different types of cancer that I won't present here. So in the next video, what I'm going to show you is in vitro, how does sickle cell anemia develop and cause the havoc that it does. For example, in patients who are genetically predisposed to sickle cell trait, in the deoxygenated state of the blood, a healthy disc-shaped red blood cell, because of polymerization and linear chain formation, becomes a sickle shape. And if you look at this video, this demonstrates by controlling the oxygen partial pressure, you can induce sickling in the lab in a chip, in a little computer chip. Just 60 seconds, and you can see the shapes change, and you can induce different shapes depending on how you do the experiments, how much time you allow. You can do it back and forth, and you can look at therapeutic avenues for sickle cell anemia, such as hydroxyurea, to try to study drug efficacy in these treatments. So let me conclude with the following observations. Bringing in very different fields, mechanical engineering, physics, chemistry, genetics, microfabrication technology, gives us unique, and combined with advances in computational modeling, gives us unique opportunities to understand human diseases. Many of these things we could not have done 5 years ago. And given all of this, it provides us new ways to explore human diseases at the tiniest of forces, timescales, and resolutions. So new possibilities for understanding diseases. And the benefits, and I've demonstrated examples of this: fundamental mechanistic understanding, new diagnostic capabilities, potentially novel therapeutics, and drug efficacy assays. And of course, this is a lot of joint effort, and these are all the people who have contributed to this work. Thank you very much.